Welcome to RenewHydro's lunch webinar, and today we have the environment special. And Olivia Simmons, Grethe Robertsen, and Line Sundt-Hansen from NINA are ready to share some previous experimental work and the plans for some of the upcoming work in RenewHydro.
So welcome to this presentation. It's this RenewHydro webinar, the environmental special. My name is Line Sundt-Hansen. I'm a researcher at NINA, and I also in charge of program 3 in RenewHydro. But today, that's not my role here. I'm a researcher presenting on the topic fluctuating water flows and warming waters in regulated rivers, understanding risks to salmon early life stages. And together with me will always also be Olivia Simmons and Grethe Robertsen. And hydropower regulation is quite common, and fluctuating flows is also quite common, and it's also natural for rivers to have fluctuating flows over seasons, weeks, and sometimes within hours. It can be quite big changes, but in some rivers, or in quite a few rivers, you have fluctuations that by far exceeds the natural patterns. And as you can see in the figure to the right, is an example of different types of what we call hydropeaking. And hydropeaking is a result of meeting electricity demand by generating peaks in the power production at a sub-daily basis. And for the normal consumer of electricity, they might not think about the impact it actually has physically in a river. But it does have an impact. And there's been done quite a few studies showing that hydropeaking does impact rivers. It can be decreased habitat quality, increased riverbank erosion, decreased biodiversity, animal drift, or animal stranding. And for fish, that actually means being left high and dry, no water. If the water disappears, or you're stuck in a pool and suddenly the water disappears. And this picture here shows an example from Norway where the water sort of disappeared and quite a few salmon were left stranded. So it's actually it's a problem, and it's something we're looking into both to study, but also maybe to solve and find mitigation measures. And hydropeaking is expected to become more widespread in the future as the demand for renewable energy increases. Hydropower provides flexible electricity production, and so we will see more of it, most likely. But it's not the only threat to life in regulated rivers. Climate change is also happening as we speak. And this figure here is from a study back in 2013. But we still see the same in the most updated studies, that over time there will be more rain and less snow, larger rain floods, more dry periods, and higher temperatures. So that's why we've chosen, as you will see when we continue, to focus on hydropeaking and warmer temperatures and looking at this combined. And just for those of you who are not salmonid people, here it's important to understand the salmon life cycle. And as you see here, you have the first phase of the Atlantic salmon life is in freshwater, the blue part. Eggs are put in the gravel by the spawning adults. Then they become alevins, or yolk sac fry. They're still in the gravel and have this little yolk sac which gives them food. But then at a certain time, when they consumed all the yolk, they need to start feeding for themselves. And this stage is called the swim-up stage. They go from the gravel and enter the river and the water. And this is a very critical life stage. Then they become par, juveniles. And when they are par, there's actually a very funny thing that can, like a alternative life history strategy. Some of these very young males have the alternative to become sexually mature as juveniles, which some of them do. And this, like the ratio of mature males differs from river to river and year to year. Then they become smolt. They get ready to go into the ocean, and they go into the ocean as smolts and becomes adults, and they grow large. They stay in the ocean from a year to up to 5 years, or more commonly 3 years, and then they come back to the same river as they were born in. But in these studies that we will present today, which is in the rivers, we will focus on the very early life stage, as they are bottlenecks for the whole population. There's a lot of mortality happening at the exact phase where the fry enters the river and starts to feed, or the juveniles starts to feed. So this is a very important life stage, and they can be impacted by water flow, temperature, but also other things like predation. And also we are doing this experimentally. So you might ask yourself, why are we not going into nature to study this, as it's happening a lot? Well, we do also study this phenomena in nature, and it has been quite a few studies from nature or from the field of what happens. But what you don't see in the field, or it's very hard to pinpoint the exact underlying mechanisms when you observe something. So by doing an experiment, we can determine underlying mechanisms, driving patterns in the field, and we can also manipulate water level and temperature and reduce the risk of confounding factors. So today we will present 3 experiments evaluating effects of rapid flow fluctuations and temperatures on juvenile salmon. We will look at swim-up timing under varying flows and temperatures, impact, that was the first one. Second one is the impact and carryover effects of varying flows and temperatures at the part stage. And the final study is growth and timing of transitions between important life stages. As I said, we've been focusing on this very early life stages, as they have a big impact on the population as such, on the population dynamics. The first experiment that will be presented today is on eggs and alevins. They start at the egg stage and end up at the swim-up stage, which I explained earlier. The second experiment is at the past stage, very early, at the very early par stage, just after they enter the river. And we looked at this stage. And then the third experiment is also at the par stage, but a later stage, later in the summer and later in the autumn. And with that, I will give the word to Olivia. Press this button when you're ready.
Yeah, great. Thanks, Lena. And hi, everybody, again. So the first experiment started at the egg stage and went to the swim-up stage. And what we wanted to know was, how do water level fluctuations and warm temperatures affect the survival between these 2 stages, as well as the size that the individual fish attain when they do this swim-up transition, and the timing of that transition. And the importance of survival, I guess, is pretty straightforward. You want your fish to be alive and not dead. But the size is also really important, because this impacts subsequent survival. So the bigger you are when you have swum up and entered the river, the more likely you are to continue to survive and thrive as a young fish. And timing is important, because you want to get the timing of this transition between these 2 habitats correct, so that the resources that you need to grow and to survive, like food resources, are available when you enter that new habitat. So how did we go about this? We used these flumes at our research facility at IMS, which you can see in the picture on the left. There's 2 flumes shown there, and each flume had a different treatment. So the first treatment, we had normal temperatures. These sort of followed the same pattern of water temperatures in a nearby salmon river. And we had stable flow. So the water would enter in the inlet of the flume and exit at the outlet, and the water level, the water height, stayed the same. In the second treatment, which was in the next flume, we had the same sort of stable water level, but this time we increased the temperature by 3 degrees. So it was warmer, a high temperature treatment. In the next flume, the next treatment, we again went back to having the normal temperature, but this time we had this fluctuating flow. So to do that, we would turn off the water at the inlet, allow it to drain, and then turn it back on. And we note that this isn't exactly the same as the way hydropeaking works, but what we were really interested in was not sort of drying out the fish and killing them completely, but understanding how this act of sudden dewatering impacts these very young fish. And in the final treatment, we had the high temperature again, so the 3-degree increase over the normal temperature, and with this fluctuation. And that fluctuation in those two last treatments happened once a day. So I'll move on to some results, and we don't have heaps of time, so I'll pick the most interesting and most relevant stuff. If you look at the figure, the bottom panel, this B panel with the purple and green points, shows the predicted survival rate under the different temperature and flow treatments. On the vertical axis, a predicted survival of 0 would be that everybody died, and of 1 would be that there was 100% survival. And what we see is that under normal temperatures, survival was much higher than under the high temperatures. So the high temperatures were bad for these swim-up fish. Within the normal temperature, we also found that survival was highest in the stable flow treatments. So when you didn't have these fluctuations, and survival was lower when you did have these fluctuations. So it suggested that these fluctuations were also bad for their survival. The high temperature treatment had a slightly more ambiguous impacts that probably need to be looked into a little bit more. So the signal wasn't as clear there how these flow treatments were affecting survival rates. The next question we had was how our treatments impacted body size or mass. And again, we can focus on the bottom panel, which has the purple and green dot. And what we found was that temperature didn't really impact how large these individual fish were at the time of swim up, but our flow treatments did. So the stable treatment fish tended to be larger at swim up than the fish from the rapidly fluctuating treatment. And the final question was about emergence timing or swim up timing. And this was a really important question because, to the best of our knowledge, it's the first time somebody's looked at how sort of rapid dewatering events can impact the timing of this transition for these young fish. And so on the figure, on the vertical axis, we have the swim up date, which as that number increases, that's later in the year. And we found that the high temperature fish swim up earlier than the normal temperature fish. But within both of these temperature treatments, the stable flow fish emerged a little bit earlier than the rapidly fluctuating flow fish. So what this means is that these dewatering events actually delayed swim up for the individuals that experienced that. And so that's a bit quick, but hopefully there'll be time for questions at the end if you have any. But the take-home message is that the water level fluctuations led to lower survival rates, smaller fish, and it delayed the timing of swim up, while the high temperatures led to lower survival and earlier swim up, but didn't seem to impact the size of the fish that much. And of course, the importance of this is that if we understand both lethal, so like mortality and survival, and non-lethal impacts like the body size and the timing of swim up, we can better develop mitigation measures. And like I said, if you have questions at the end, you're welcome to ask, but we also have published this work, so you can see the citation there in the slide. And I'm going to hand it off back to Lena.
Yes, it's the chair game today, so it's me again. I'm presenting the experiment number 2, which is, because since we're going chronologically, we're at the par in early summer. And as I said earlier, we were looking at impact and carryover effects of varying flows and temperatures at the par stage. And we tested impact of warm temperatures in these stream channels, which you see below here. It's like narrow stream channels, but it's very good for juvenile salmons and allows us to manipulate both water flow and temperature, which we did. I will get back to that shortly. And then when we've done that for approximately 6 weeks, we transfer them to a more natural system where there were natural predators and feed, which is called the river park, which you see here below. And we measured growth, survival, and what we called carryover effects. And carryover effects are, or that is when an organism experiences something in one life stage or in a season, and this will affect the performance, behavior, or survival in some subsequent stages or seasons. So our idea was that perhaps the impact of warmer temperatures and fluctuating water levels would maybe not be visible directly, but then perhaps it can be visible at a later life stage, which we try to measure. And I will also say this is the last experiment to be performed. It was finished last autumn. So we still don't have all the results, but I will get to this. So in the first part, as I said, testing impact of fluctuating water flows and temperature. The temperature were approximately 3 degrees above the normal temperature at all times, which you can see on the panel to the left. And then something a bit strange happened here. It shows actually it's a time-lapse video of how we reduce the water flow. But what I can say is that the water flow was reduced until it was just like barely water there. So we kind of more testing the impact of the stress of the fish when water is disappearing together with differences in temperature. And at the end, as I said, we transferred this fish after a month or so of experiencing this treatments to a more natural-like river park where they were for another month. And we are still analyzing data material. What I can say is that we saw good recapture rate. Thus, we didn't have a very high mortality rate. We can already say that we saw some growth differences as a result of the temperature differences. But here are more results to come, so I will just say to be continued. And then I will give the word to my third colleague, which is Grethe, who will take my chair as we have our technical problems. Grethe?
Okay, thank you, and thanks, Lina. Yes, I'll present some results from a third experiment that we performed, which follows in time after what Lina presented. So this was done slightly later in the season. It's still on the young of the year stage, so it's done with salmon that hatched the same year as we did the experiment. And it lasts from late summer to autumn. And the main question that we asked is, again, like how does the stress associated with water level fluctuations impact the salmon? So it's more like the perceived stress and not that they're drying out or anything. And how does it affect the survival, growth rate, and also the timing of key life history transitions? And an example of such a life history transition could be timing of sexual maturation in young of the year males. And I couldn't find a picture of a salmon, so here I'm showing how we found, how we can see if a young of the year male is sexually mature without actually killing it and opening it up. And that's to look at whether it expresses milt or not in the spawning season. And the predictions, well, some of the predictions that we had was that fluctuating water levels during the early development may act as a stressor and thereby suppress growth, which would result in smaller body size at age during a time when the body size is a key determinant of survival and the timing of developmental transitions. And I'll come back more to this later. So for the setup of this experiment, we used 3,400 individuals that we pit marked. And for the ones of you that don't know what a pit mark is, it's kind of a radio transponder that is within a glass cylinder, and it contains a unique code. And then we place this kind of tag inside the body cavity of the fish, and so we can follow them throughout their life. So we started the experiment by pit tagging them, and then we recorded the length and mass of all the individuals in mid-August. And after that was done, we placed the fish in different fish tanks with 2 different treatments. So one was, again, was stable water levels versus fluctuating water levels. And we let these treatments continue for 2 months to make sure that it's over a long enough period so that we could see an impact. And the way it worked was that we lowered the water level 1 time per day down to 1 centimeter above the fish. And we did this at unpredictable times to avoid habituation. And we ran this 5 days per week. And after a while after we ended the experiment, we also re-measured length, mass, and also checked for sexual maturation in the spawning period of this young of the year salmon. And the results of this we used to determine whether just the pure stress of experiencing near dewatering affected survival or growth rate, and also the proportion of sexually mature young of the year, so the ones that actually decided to mature sexually. Based on the results, we actually didn't find any treatment on growth rate or survival, which was actually a bit surprising, we thought. But we did find a effect on of the treatment on the proportional part that matured sexually. So there was a higher proportion that matured sexually, the fluctuating water level treatment, than there was in the control, so almost double as many. And I'll take you through the figure. So if you look at the x-axis, there's an impact of initial mass, where in the stable water temperature treatment. So the higher the slight impact. So where the fish that were larger individually had a slightly higher chance of becoming sexually mature par. In comparison, in the fluctuating water level regime, there was a much steeper slope between the initial mass and the probability of an individual being a male sexual mature par. And so based on that, it seems like early life exposure to stress associated with fluctuating water levels can actually modify the life history trajectories without detectable effects on survival or mean growth. As the treatment altered, the fluctuating water levels altered how body size translated into maturation probability. So this is in line with that there being a state-dependent response to a perceived environmental risk. So means, given that the fish were exposed to such a harsh treatment over a period, they probably perceived the long-term survival prospects to be poor, and thereby kind of went with a strategy that's kind of best of a bad job. So it's better to mature early and have one shot at reproducing than to die before you have a chance to reproduce at all. And one of the reasons this kind of change in that more individuals became mature earlier is important is because early sexual maturation can actually delay the timing of seaward migration, which could also decrease the survival both in the freshwater stage, but also in the saltwater, like after they go to sea, like a type of carryover effect. We have actually released this fish to sea, and they have come back, but we haven't managed to analyzed the results yet. So over to Olivia again.
Yes, my turn. So to sort of wrap things together a little bit, what we've done so far is tested the impacts of these fluctuating water levels, as well in some cases temperature, on various juvenile life stages. But if you've been astute or observant, you'll notice that there's something missing. We started the first experiment with the eggs and went up to swim up, but then the next experiment started about a month after swim up. So there's sort of a gap in our timeline for the experiments that we've run so far. And as Lena mentioned, this is a particularly important period, this phase just after swim up, because mortality rates naturally tend to be quite high. So if there's a stressor like fluctuating flows or high temperature during this stage, it could exacerbate those naturally high mortality rates. And so in RenewHydro, we're planning a fourth experiment. And the point of this experiment is exactly to target that particularly vulnerable part of the life cycle. So what we want to do is we want to assess the impacts of flow fluctuations on survival just after swim up. We're going to start at the alvin stage. And we've got a photo on the slide of some alvins. So you've got a little bit of an idea of what these little fish look like. This experiment is only part of RenewHydro Work Package 2.2. And we're planning on running the experiment in spring 2027, so spring of next year. And we're really excited because we're going to be collaborating with some other people within the same work package that are also working on hydropower related topics. PhD student Magnus Graf and Professor Knut Alfredsen at NTNU, who are working on understanding ways to mitigate the environmental impacts of hydropeaking. And so we're going to team up to plan this experiment together. We're planning on using the stream channels as well as the river park, the two facilities that Lena presented. And we're actually going to try to conduct the sort of peaking or the fluctuating flows in the river park this time in that sort of natural setting. And our predicted results is that we'll see lower survival in the fluctuating flow because we know this is a critical period for survival, but we'll test other things too. For example, we'll look at growth rates and see if that has also decreased in the fluctuating flow treatment.
Hi, would you like to join this meeting now?
So to sort of try to paint the whole picture of what all these disparate experiments can tell us, what we're seeing is that the impacts of warming climate and hydropeaking on juvenile Atlantic salmon are likely to vary by life stage. So some life stages might experience non-lethal impacts, which we saw in the first experiment when the fish were a little bit smaller, or they were delayed in their emergence timing, or in the third experiment where the fish that were sort of stressed.
Something went wrong. Please try again later.
Were kind of stressed by the fluctuating flows, matured early. These are sort of non-lethal impacts that could have negative long-term consequences for the population in terms of the population demography, but also individuals in terms of their life histories. And then, of course, other life stages might actually experience lethal impacts, so alterations to their survival rates, which we saw in the first experiment and which we anticipate seeing in the fourth experiment. And if we can better understand how these impacts vary through the life cycle, then we can improve mitigation efforts by knowing when in the season to sort of target mitigation efforts to make the most effective. So that's everything from us. Thank you very much for your attention. If you have any questions, we're happy to take them. Our contact info is also displayed on the screen. Yep, that's it from us.
Yeah, thanks.
Thank you very much, all of you. Now we do have time for some questions, so just raise your hand.